The internal structure of the kidney includes the outer cortex and the inner medulla. The ureter divides into branches and ends of which envelope medullary tissues, which are called the renal pyramids. Look at the renal pyramids here, the yellow ones. The actual work of the kidney is carried out by functional units called nephrons, and each human kidney contains about 1,000,000 nephrons. This is the ultimate functional unit of the kidney, the nephron. Each nephron consists of vascular and tubular components. An afferent arteriole, you can see the afferent arteriole, carries blood to a knot of capillaries, and this knot of capillaries is called the glomerulus. Draining each glomerulus is an efferent arteriole that gives rise to peritubular capillaries, most of which surround the cortical portion of the nephron and the tubules. These are the peritubular capillaries. And now we have got the cortical part of the kidney and the medullary part of the kidney, and we have got the proximal convoluted tubule, the loop of the Henle, and the distal convoluted tubule, and the collecting ducts, where many of the DCTs join to form the collecting ducts. We will see these details a little more. Blood pressure forces water and smaller molecules to be filtered from the glomerulus into the Bowman's capsule. The initial segment of the renal tubule is called the proximal convoluted tubule, and the glomerulus and Bowman's capsule and the proximal convoluted tubule of each nephron are located within the cortex of the kidney. And now, from the proximal convoluted tubule arises the loop of Henle. The nephron tubule turns down into the medulla. This portion of the tubule in the medulla is called the loop of Henle, and this has got a descending limb of the loop of the Henle, the axial loop of the Henle, the narrow portion of the ascending limb of the loop of the Henle, and the thicker portion of the ascending limb of the loop of the Henle. And this loop of the Henle terminates in the distal convoluted tubule, which further drain into the collecting ducts. Remember, the collecting ducts are again in the medulla. The entire loop of Henle, the descending limb, the loop, the ascending limb, the thick portion are all housed within the medulla of the kidney. Now, the distal convoluted tubule of many nephrons join into a common collecting duct in the cortex, and the collecting ducts then run in parallel with the loops of Henle down through the medulla and empty into the ureter at the tips of the renal pyramids, which we have seen in the previous diagram. A few peritubular capillaries run into the medulla in parallel with the loop of the Henle and the collecting ducts and form the vasa recta. You see, these are the vasa recta. So, the loop of the Henle is surrounded by few peritubular capillaries which run down into the medulla, and these capillaries carry away molecules that are reabsorbed from the tubules. All the peritubular capillaries join back together into a venule that eventually leads to the into the renal vein. So, the loop of the Henle, the descending limb, the vasa recta, and the ascending limb, and then the venule and the renal vein, and of course, this is the renal artery. Now, nephrons regulate the composition of the blood and urine by a combination of three processes called the filtration, secretion, and reabsorption. Filtration occurs from the glomerulus into the Bowman's space or the Bowman's capsule, and then the fluid passes through the PCT and then the descending limb, the loop of the Henle, then the ascending limb, and then the distal convoluted tubule. During the process of the passage of this ultrafiltrate through these regions of the nephron, there will be most important step will be the reabsorption of the filtered fluid. I told you 180 liters of fluid is filtered, out of which only 1.5 liters has to be excreted. That means about 98% of the fluid and majority of the molecules like glucose, sodium have to be reabsorbed back. So, reabsorption is a very important step in the kidney. Then of course, certain ions and certain molecules are secreted for excretory purpose into the urine when the urine passes through the tubule and its segments. Viewed schematically, we will see how these processes are facilitated by the regular arrangement of the segments of the nephron. Now, these are the vasa recta, and this is the descending limb, and this is the thin ascending limb, this is the thick portion of the ascending limb, the DCT and the collecting duct. Remember, all these are housed in the medulla of the kidney. The Bowman's capsule and its glomerulus are responsible for the first step of urine formation, that is the filtration of urine. As blood flows through the glomerulus, pressure forces water and smaller molecules out of the glomerular capillaries into the Bowman's capsule, and the basement membrane is the only layer of membrane between the capillary layer and the Bowman's space. This filtration process leaves the blood cells and the proteins behind in the efferent arteriole. So, important hormones and molecules which are not to be filtered are protein bound, so that they escape filtration at the Bowman's capsule. Now, let us see what happens. As you see here, the glomerular capillary brings the blood here, and the urine is ultra filtered, and this is called the primary urine. 180 liters of primary urine is generated, and then the proximal convoluted tubule is responsible for the excretion of sodium into the peritubular space, and sodium is reabsorbed very quickly in the proximal convoluted tubule into the medullary region. Okay. The proximal convoluted tubule is responsible for most of the reabsorption of water and solutes from the glomerular filtrate. The cells of this section of the nephron actively transport sodium, with chloride following passively. When the sodium molecule goes, chloride molecule passively goes, and it is reabsorbed as NaCl. The active transport of solutes out of the proximal convoluted tubule into the tissue fluid causes water to follow by diffusion passively in order to maintain the osmotic gradient. The water and the solutes moved into the tissue's fluid are taken up by the peritubular capillaries and returned to the venous blood that leaves the kidney. And now, despite the large volume of water and the solutes reabsorbed out of the proximal convoluted tubule, the ability of the kidney to produce urine that is hypertonic to the blood plasma is due to the loop of the Henle. The loop of the Henle does not concentrate the urine directly, rather it functions as a countercurrent multiplier. Okay, you see the increasing concentration gradient from the cortex into the deeper medulla. It functions as a countercurrent multiplier, creating a concentration gradient in the surrounding medulla. Now, to understand the countercurrent multiplier mechanism, it is easiest to move backward through the tubule, starting with the thick ascending limb. The thick ascending limb actively transports chloride and sodium. The thick ascending limb is this portion, and that actively transports chloride and sodium into the peritubular medulla, and from the tubular fluid, the sodium chloride moves into the surrounding tissue fluid. And now, the thick ascending limb is not permeable to water. Remember, no water is reabsorbed in this part. So, the reabsorption of sodium and chloride out of this part of the tubule is not followed by outward diffusion of water, unlike here. This reabsorption of sodium and chloride raises the concentration of solutes in the surrounding tissue fluids. Okay? Now, the concentration of the solutes is raised in the surrounding tissue fluid in the medulla, because only the solute is reabsorbed and not the water in this part of the ascending limb of the loop of Henle. Now, the descending limb in contrast is permeable to water, but not very permeable to sodium. Since the surrounding fluid has been made more concentrated, water leaves the tubule by osmosis. From here, water leaves the tubule by osmosis, as you can see here, because there is a higher concentration here, and this is permeable to water, and passively water is thrown or reabsorbed back into the surrounding medulla. As a result, the fluid in the descending limb becomes more concentrated as it flows towards the bottom of the medulla. So, from here the sodium is reabsorbed here, passively water follows. Here water alone goes because there is a lot of sodium that is coming from the DCT, and to match that there is water reabsorption. So, slowly the urine is getting concentrated, and when it reaches to the bottom of the tubule in the medulla, the urine becomes more concentrated than what it is here in the proximal convoluted tubule. Now, the thin ascending limb is not permeable to water. This part is not permeable to water. It is, however, permeable to sodium and chloride. Since the tubule fluid is more concentrated than the surrounding tissue, sodium and chloride diffuse out. The thick ascending limb continues to move sodium and chloride to the medulla by active transport. Now, you see here sodium and chloride are moved out because this portion is comparatively less concentrated than that one, and so sodium and chloride move here, and here also sodium and chloride is reabsorbed. Now, as a result of this process, the tubule fluid reaching the distal convoluted tubule is less concentrated than the blood plasma because more of the solute has left, and this fluid is dilute compared to the plasma, and then the solutes that ever been left behind in the renal medulla have created a concentration gradient in the surrounding tissue fluid. Now, this results a further increase in the concentration of the solutes in the lower part of the medulla. As a result of this process, the tubule fluid reaching the distal convoluted tubule is less concentrated than the plasma, and the solutes that have been left behind in the renal medulla have created a concentration gradient in the surrounding tissue fluid. And now, since the fluid entering the distal convoluted tubule is less concentrated than the surrounding cortex, the tubule loses water osmotically as it flows through the collecting ducts. Now, here is a lot of concentration of solutes here, and the dilute urine has come here. So, more of water is available, and that water is now reabsorbed back into this one at the DCT, and also next in the tubule fluid entering the collecting duct is at the same concentration as the blood plasma. By the time it comes here, it will have the same concentration as plasma. However, since sodium and chloride have been moved out of the tubule fluid, urea and other waste products make up for the greater proportion of the osmotic pressure and the solute content of the fluid that is in the collecting duct. And now, as the collecting duct descends from the cortex to the tip of the renal pyramid, the concentration gradient established by the loop of Henle increases, and this increasing solute concentration causes more and more water to be absorbed from the fluid, thus concentrating the urine in the collecting duct. Now, you can see how the urine is being concentrated. It is coming down, and more of water is being reabsorbed. Remember, this reabsorption of water is under the control of antidiuretic hormone, whereas the water reabsorption here it is passive because sodium chloride diffuses. Here also it is passive because there is higher amount of surrounding tissue concentration of solutes, and here there is no water is reabsorbed at all because it is impermeable to water. Here whatever water is reabsorbed again is passive, whereas here the water reabsorption is active, and it is facilitated by the antidiuretic hormone or the posterior pituitary vasopressin. In the absence of vasopressin, water cannot be reabsorbed in the collecting duct, and that results in a dilute large quantity of urine, which we term as polyuria. So, this is in a nutshell the collecting mechanism of the urine right from the Bowman's capsule into the ureter, and we have got the concentration gradient, the increasing concentration gradient across the medulla, the counter current multiplier and the counter current exchanger systems, the passive reabsorption of water in the PCT, the passive reabsorption of water in the descending limb, the non-permeability for water in the ascending limb, again the passive reabsorption of water in the DCT, and again the active reabsorption of water in the collecting duct under the influence of ADH. This in a nutshell is how water and sodium chloride is handled in the kidney right from the Bowman's capsule, glomerular filtrate up to the point it reaches the renal pyramid. Of course, there are many other ions like potassium, urea, and then other molecules whose mechanisms of absorption and secretion are not discussed here, because our subject is about polyuria and we are more interested in knowing how water is reabsorbed in the renal tubules, both in the proximal and the distal convoluted tubules and in the loop of Henle. So, with this I close this animation and we get back to our continuation of the lecture.